So in this lecture, we are going to discuss another characterization technique for carbon materials, electron microscopy. Our focus is going to be on transmission electron microscopy because this is the microscopic technique which is used for looking at the microstructure. You have, in fact, I have already shown you a couple of transmission electron micrographs or transmission electron microscopy images. So I have shown you before also to help you visualize how your carbon material looks like at a very, very small scale, at an atomic scale. How are the crystals organized? Although it is very powerful tool, we can visualize, looking at whatever you have made gives you a kind of satisfaction, right? Okay, fine, I wanted to make carbon nanotubes. Here are my carbon nanotubes. So when you see them, that does really help you understand your material. But at the same time, there are few small things that we need to take care of, because in the transmission electron microscope, you may sometimes also get some wrong impression of the material. So this is what we are going to discuss. So similar to the other techniques that we have discussed, I'm not going to go into the fundamentals of transmission electron microscopy. I will give you references and resources to learn about that. But we will be focusing on how does it work for carbon, especially these confusions, right? When some planes look like they are on top of each other, but they are actually not. Some planes look like they are intersecting each other, but they are actually not. So all of this we are going to discuss. One important thing is that, and that I will probably again tell you in this lecture, is that TEM should not be your only analytical tool. So whenever you've made a carbon material, you kind of, even if you know what material you're making, whether it is nanotubes or graphene sheets or a bulk carbon, in all cases, TEM should be more like a supporting technique, and that should not be the only technique. X-ray diffraction often gives you good average of the crystal structure of the entire material. And in the case of transmission electron microscopy, you're often looking at a very small region. And there also, the interpretation is not all that straightforward. This is what we are going to discuss. Okay, so before, I will not give you too many details, but let's at least understand why and how we can use electrons for imaging something. Okay, so often you would image something using light, or even you can use X-rays to get images, like in the case of your bones and your body. So you can use different electromagnetic rays for the imaging purposes. But can we also use electrons? Because electrons, we don't know what they are, right? Whether they are waves or particles, we don't know. However, there is one thing that definitely works for electrons, is the de Broglie relationship. So de Broglie, that is also pronounced de Broglie. This relationship you definitely know. Lambda equals h by mv, where h is your Planck's constant, and whatever is your particle. Let's assume that electrons are particles. In that case, the mass of those particles is your m, and then v is the velocity at which they are accelerated. So this is something very fundamental. Now, if I take electrons that are accelerated, let's say by some voltage v, in that case, this is you also understand this is very simple. To square root of 2 MeV, that is what you will divide it. So that will be because these are accelerated particles. So this is what your relationship changes to. Okay, and again here you have two new parameters. So m, now we know what is the mass of our particles that we are dealing with. So that is, you know the mass of electrons, you know what is the charge on electrons. So these are the two more things that you need to factor in. Okay. However, what is interesting is this relationship is only valid for electrons that are low energy. Low energy means the voltages at which, using which we are accelerating them are not too high. And we will see what do we call too high, what is not. So these are low energy electrons, and for those, this relationship is valid. Why? Because we are sort of ignoring any relativistic, factors. So what happens if we have these very high energy electrons, and we also need to make relativistic considerations? In that case, this is the relationship. This is what it changes to. Okay. So now, using all of these relationships, you can calculate what are the wavelengths of electrons that are accelerated using different voltages. And here I have shown one table in which you can see. So the left two columns are relatively low energy electrons which we use for scanning electron microscopy. So scanning electron microscopy we are not going to cover in this lecture, but that is something which is mapping your surface. So that is just taking the image of your structures of your surface, but not going through your material. So transmission, as the name itself suggests, there is some sort of transmission of these wavelengths through your material. So they are transmitting and we are collecting. So you have some, you have this, this is your sample, the electron comes from here and then it goes through your sample, comes out with the loss of some energy also. So that is what we know as transmission. And for transmission electron microscopy, the wavelengths that are, in the second column of this table, these are the ones that are used. So now from what do you see here? The wavelengths associated with electrons, they are in the picometer range. So they are, much shorter than even your X-rays. So if you remember in the X-ray diffraction lecture, I said that we were talking about the lambda or the wavelength should be pretty much of the same size of the interatomic distances, and that is how we use that is why we use X-rays for diffraction purposes. What if your wavelength is even smaller? Then you can very clearly see your interatomic distances. You can even potentially see single atoms. Although this is not the case often because of the instrumentation parameters. But in principle, you should be able to see single atoms using transmission electron microscopy. And there have been reports where people have observed, the hexagonal rings of graphene. Although, again, as I will explain to you, the interpretation can be tricky. Sometimes also these beams of electrons can damage your sample because after all, electrons do have certain mass. So they might also damage your sample. In that case, while imaging your sample, you may end up getting, inducing defects. And in that case, you will not be sure whether the defect was already there in the material or it is because of what you did to the material, right? So all of these things need to be factored in. But you see that the wavelengths are in the picometer range. That means the imaging is not really wavelength limited. So we have, in terms of wavelength, we have a lot of flexibility. We can go all the way to picometer region. Okay, so I already said this. You have much shorter wavelengths than X-rays. Okay. Now, electrons, therefore, are used for getting microstructural information. Remember that now we are talking about microstructural information. We are not talking about crystal information. Although microstructural information does contain the crystal structure, and you remember that you also can now see the defects, the residual stress inside your material. So now you can actually visualize all of these things. But now we are more concerned about the microstructure, not the large scale crystallinity of the material, although we can deduce it from here as well. Okay, fine. So only the high energy electrons are used in transmission electron microscopy that you already saw in the table before. Okay, now here I have shown some schematics. So I will again not go into the details of what kind of lenses are used and what is the optics of a transmission electron microscopy, but this is some basic schematic of the entire instrumentation. And here you can see that this is a very long structure. So if you see one transmission electron microscope in some lab, then it looks like a tower. So it is a tower-like structure, 6, 7 or more feet higher. Okay. So these kind of towers, they contain various lenses, and these lenses are not like your, what you would think of, optical lenses and so on. These are electrostatic and electromagnetic lenses. So these are specialized lenses. Okay, and then you have an electron source. What you need, the most important thing for you is an electron source. So you will use some thermionic source. So in some other lecture I had mentioned thermionic source is when you heat something and therefore the electrons come out. Okay, so you heat something and you also create a high potential difference, then the electrons come out of the source. So these thermionic sources, although there are many other more sophisticated sources, but you could potentially just use a tungsten wire. In both scanning electron microscopy and also transmission electron microscopy. You also have other materials like lab 6. There are other materials, but you have basically one electron source. Now you have the beam of electron which is focused through these lenses. Then it falls on your sample transmits through your sample, and in the bottom you have a screen or a detector where you form the image. So this is the fundamental principle. Okay. And yeah, so basically now you're using a beam of electron for imaging purposes. However, you should know that not all the electrons will just transmit through your sample. And so there may be some back scattering and so on. And also definitely some electrons will damage your sample also. And definitely even those which are transmitting through your material will lose some energy. There will be some interaction, some loss of energy when your electrons are going through your material, because they are after all interacting with the atoms of your material, right? So there will be some loss of energy. So it's these electrons that would lose their energy. Okay, so this is the fundamental principle. Okay, when you prepare the sample, so now we already come to more of the experimental part and the analysis part, right? When you prepare the TEM samples, in principle it should be thinner than 20 nanometer. If you want that the electrons pass through it, and if you want to see the microstructural information. If you have a thicker sample, what happens? Well, nothing bad will happen. The only thing is that you will not be able to see the microstructure. Why? Because if your sample is too thick, then the electrons, they sort of get lost inside your material. They don't come out of the other side. And even if they come out, they have lost a lot of energy. So what you will see, you will lose a lot of information in that sense. You can definitely, evaluate if the diameter of your tube, let's say, was 50 nanometer, or if you're using a carbon fiber. Well, often I see transmission electron micrographs of carbon fibers, which are even 200 nanometer thick, okay, or their diameter is 200 nanometer. In that case, well, you can measure the diameter of your fibers, but that you could even do using scanning electron microscopy, right? 200 nanometer, in fact, even there are optical microscopes that can give you that information. So for that purpose, in principle, there is no need to use transmission electron microscopy, especially high resolution. So as the resolution increases, we are basically using higher and higher energy electrons. So that information can also be achieved by other techniques. When you want to essentially get the microstructural information, you want to see how your crystal planes are organized. In that case, the sample should be thinner than 20 nanometer, and the thinner the better. So if you have a sample that is 5 nanometer thick, that will give you more information, because you will have only a few planes of your sample inside your within the focal length. So this is something we are going to also discuss. The point is that you need to spend some time in preparing your TEM sample. Okay, and that is why also it becomes a little bit tricky when you're using let's say bulk carbon materials. If I want to take the transmission electron micrograph of a glass-like carbon material, and I purchase glass-like carbon in bulk. Okay, in that case, what I will need to do is I'll need to make either a powder or a slice, which is thinner than 20 nanometer, which you cannot use just some kind of milling or ball milling or something like that to make that kind of sample, because 20 nanometer, believe me, is very, very small. Okay, so you will have to maybe prepare a slice using what is known as focused ion beam milling. Okay, we will not again go into details, but as the name itself suggests, there is an ion beam which can be used, which is a ion beam is high energy beam, which can be used for slicing your sample, and that also needs to be done under inside a microscope, because you should be able to see what you're doing there. Okay, so often these are the scanning electron microscopes are also equipped with this ion beam setup, or you can buy a separate focused ion beam setup. There you can actually slice your sample and then get this very thin, ultra thin sample. So you also know that you might be inducing some stresses in your sample. Or if you're using any technique for making powders, in that case also you need to make like really thin powder. So the sample preparation is not all that easy. Okay. Now, can you get only images from transmission electron microscopy? No, there are many things that you can do with this machine. But the most commonly used form, or the most common application of transmission electron microscopy is imaging, because as I said, it helps you visualize your material. That is very good, but you can also get diffraction patterns, electron diffraction patterns, like because So you already now know about X-ray diffraction patterns. The only thing we are doing here is changing the wavelength, but we can also get the diffraction patterns. The diffraction patterns are often in the reciprocal space. So that is why you can get a more global information, because then you're getting the reciprocal, right? So it's like for the entire sample, you can get the information which is sort of localized. And so while imaging will give you more local information, the diffraction patterns can give you relatively global information. It does not help us very much with non-graphitizing carbons. So again, we are going to discuss that. Non-graphitizing carbons, you know that they are turbostratically arranged and their sheets are in, they contain defects and so on. In that case, there is what is missing is the 3D crystal structure. So we have crystals in 2D, but 3D arrangement is missing. And whenever there is 3D arrangement missing, it becomes very difficult. Even with XRD, it is difficult. But potentially you can get electron diffraction patterns as well using your transmission electron microscope. Okay. Yes. So this I already told you. The images cover a very small space. Typically, what you're looking at is this information within 50 nanometer, of your sample. So remember that if your sample is not uniform, sometimes it happens. This has happened to me that I thought I found something really amazing in one corner of my, of my material, but that was really just one corner and not the rest of the sample. And it could even have been some impurity. Certain microstructure can also come from, let's say, if you have some non-carbon atoms in your material. So they may change the shape of the how your 6-membered rings are organized. So because of defects, you can have different strains in the sample. So because of that, often if you're sampling only one part of your material, then you may end up getting misleading information. So you need to often perform the microscopy at different samples. You need to do the sampling at different positions on your sample, on your specimen. Okay. So yes, one more thing is that you can plot the local intensity profile. So I'm mentioning this because this is also very commonly seen in a lot of publications and so on. You will see these, what is called intensity profile. I'll show it to you in the next slide. So intensity profiles are basically, this is just something done by the software. So this is not really a part of your, even if you can just take a TEM image and you can do this processing by yourself using practically any image processing software. So what you're doing is you're taking the profile, if you have a dark line and then bright and then dark and so on. So what you want to see is the spacing between them. So you will just take the peaks or the profiles as the intensity increases and decreases, and that helps you understand the D-spacing. D-spacing is very important to us, and that is why often you will see intensity profiles in many publications related to carbon materials. Okay. Electrons do damage the sample, and this is something, especially when your samples are sensitive, then you need to be, and since carbon materials are sensitive, so they're definitely sensitive to, especially if you're working with these few layer graphene structures, for example. These materials are sensitive to electron beams, and you need to be careful. Also, if you're working with carbons that are prepared at lower temperatures, which may potentially contain some impurities, and they may not be perfectly electrically conductive. So you know that electrical conductivity increases as we increase the heat treatment temperature, right? So if you prepare something at let's say 700 or 800, you may not even call it carbon. That's a different thing. But let's say if I say it is a pyrolyzed polymer, for example. So in that case, these materials may not be electrically conductive, and one condition for TEM analysis is that you need to have an electrically conductive sample. So the electrons need to go through it. So you can imagine that you need to have something that is electric, that allows the transport of electrons, right? And materials that are not electrically conductive often are sputtered or coated with a conductive material. You can use any metal to coat gold, for example. Okay, that you will also in fact do for scanning electron microscopy, right? Because electron microscopies require the samples to be conductive. But you should understand that you may, if you're working with, for example, single or few layer graphene sheets, in that case, you may end up losing some information because of this sputtering. So all of these, but graphene happens to be anyway electrically conductive, so it's not a problem. But I'm talking about the defect containing or impurity impurity containing structures, graphene-like sheets. So because these kind of sheets will also exist in the material that is at 800 degrees, but they are definitely not pure carbon. So you need to be careful. Your samples are sensitive to electron beams, and the electron beam can potentially cause damage to the, so if not the entire sample, surface of your sample, but well, it's pretty thin already. Okay, so transmission electron microscopy has been used to image carbons for a very long time, since the 1950s. In fact, when we were learning about carbon nanotubes, I showed you an image of these hollow carbon filaments or carbon tubes that were observed in 1952, and those were TEM images. So TEM itself has a long history. The first the electrons were used for imaging purposes as far back as in 1930s. So 1931 we had the first TEM setup. But of course then there were various modifications, and now it has become a more common technique. Okay, the point is that if we relate the history of carbon materials development and the history of transmission electron microscopy, these are connected because we have always been using TEM to visualize our carbon materials. Okay, so as far back as in 1950s we used it. Okay. One very interesting development in the entire carbon community was the fact that we understood what are non-graphitizing carbons, right? And a lot of carbons are non-graphitizing. We know that now. Okay, that all this the discovery of non-graphitizing carbons, or the how we understood the microstructures, or when there were microstructural models proposed. Many of them were very strongly related to TEM imaging. So not the first one, not the one that Rosalind Franklin described. That was completely based on X-ray diffraction, and of course some properties of the material that you need to correlate. But the other models, the Jenkins-Kawamura model, and then the model provided by PJF Harris, and also the recent study then that I have done, and then I've shown you also the in situ electron microscopy, which gave us some more information. Then there are also, recently you will find many other publications, which nowadays we understand the microstructure of non-graphitizing carbons reasonably well. Okay. So yeah, all of these studies have been very much dependent and very much related to transmission electron microscopy. So this is a very important imaging technique for us. Okay, carbon nanomaterials. So I told you, you've never seen a carbon nanotube. So you only believe in what you see inside the TEM. Not just carbon nanotubes, but there are also carbon nanoparticles, various carbon nanomaterials. And in fact, some fancy carbon materials, fullerenes. Okay, fullerenes we know, and we know about them also chemically using chemical synthesis, and we also know how they agglomerate and then they form a FCC crystal and so on. We know what is the structure of fullerenes also because of the other characterization, other information that we have about the material. However, in order to visualize them, this is a very interesting tool, and you can even have very when I say fancy carbon materials, you have carbon nanotubes and you have fullerenes filled inside them. These kind of structures, the TEM provides, it's a very good tool to see these structures and to then understand and then correlate the rest of the data. Okay, so carbon nanomaterials definitely are, they heavily rely on transmission electron microscopy. Okay, but then till now I have only been telling you all the nice things. It is nice, everything is nice about TEM, we'll talk about that, it's a very important tool and so on. But there are also certain things that we need to take care of. So when we are interpreting the images especially, at that time a lot of things become important. That is what we'll now discuss. So these are some of the few important aspects. These two aspects I have highlighted. So two things, when you see any carbon material, let's say I have this. Yeah, so if I see this, what is going to be visible? Only the edges. Only the edges. You will not see this central part because electrons, our carbon materials seem to have a reasonably high transparency to electrons, at least the lower energy electrons. So it is only the edges that are visible. That is number one. Number two, this is not, the second one is not just valid for carbon but for everything. That transmission electron microscopy images, they are basically 2D projections of a 3D material, because your entire sample, which is your 20 nanometer or thinner sample, that is in the focal length. So what will happen when when there are two objects which are for which you cannot get a depth perception? So if I have this one sheet and this is my other sheet, now I don't know, maybe you can see this. So they are like this, they are far apart from each other, and if I rotate them, then they are like this. Okay, I hope this is clear. Now, however, what you see is just the edges, and it looks like they are both in the same plane. So what you cannot differentiate if they are like this or they are like this, basically, because you are only seeing the front view, and in the front view all you see is the edges, and you see that they are just you cannot get any depth perception. So this is something very important. These two things, if you understand, then you should be able to analyze your images at least. So the problems during imaging that are different, but once you get the image interpretation, at the time of interpretation, you need to understand these two things completely. Okay, fine. So as I said that we do have carbon materials are also transparent to X-rays and to various other wavelengths, but also for electron wavelengths, they do have a reasonably high transparency, especially for low energy electrons, but we need to use low energy electrons, because high energy electrons damage your sample. So most of the TEM equipment that are available, they often operate at 300 kilovolts, but you can go down to 100, but it's really difficult. In fact, a lot of instruments do not allow you to go below 100 kilo electron volts. So that means, and what is the optimum voltage is below 80 kilovolt. So if you want to image carbon samples, in principle, you should optimize. You should image them only below 80, because 80 kilo electron volts is the this is the energy required. Not exactly 80, but slightly above 80 is the energy that is required to remove one carbon atom from your 6-membered rings, which basically means that above 80 kilovolts you can potentially induce defects while imaging. So this is another aspect that we need to be careful about. Okay, sample preparation itself, I already told you that this can already create defects. So you are often using either this focused ion beam milling or some sort of technique for making powders. But in that case, you're definitely using very high energy to make these slices or to make the powders, and there you may also have higher temperatures occasionally, depending upon how you're making your sample. But if you have a high temperature, our materials are temperature sensitive. You know that. I mean, all we are trying to study about carbon, especially pyrolytic carbon, all we are trying to understand about these materials is that, what is the influence of temperature on the microstructure. And if you induce high temperatures during your sample preparation, in addition to the mechanical stresses, so in that case, you may end up changing the microstructure of your material. However, often it is not so significant that it will completely change. Also, if you're, as I said, you're doing the imaging at different locations, then you will get certain idea of the material anyway. So just all you need to know is that don't do very extensive sample preparation. So don't use techniques that can, that would also depend on what kind of carbon material you're using. So just be sure that you do not damage your sample, and if you do, then still try to get do the sampling at different locations and things like that, just to make sure that your data is correct. Okay. So now I'll show you how, so how does a transmission electron micrograph looks like. So micrograph is the term that we use often. You can also just call it a TEM image. Okay. So this is one example of one carbon material. This is how it looks like. Okay. How does a diffraction pattern look like? This is how it looks like. We call it selected area because it is often done in a very small region of the sample altogether. Selected area diffraction patterning. Also, we select an area in the image, and there we do the. This will be important for when your material is, some part of it is crystalline, some part is not crystalline. So, okay, these are the diffraction patterns. This is how they look like. Now, what often people do is they do this what is known as indexing of diffraction patterns. Indexing basically means you will take two planes. So what you see here, these are lines, they are planes, right? So you have one plane like this and another plane, let's say like this. What you will do is you will measure the lengths of these distances. So between two spots. So you will measure distance between two spots like this, and then also okay, two spots like this, whatever is the perpendicular. Okay. Not necessarily perpendicular, but these are the two planes. Okay, you will measure the lengths, and then you will find the ratio of these of the lengths, and accordingly for all types of structures, FCC, BCC, all different crystal structures, there exists some rules of thumb that if this is the ratio of the two, then it could be potentially an FCC. It's not that simple, but the point is that you can obtain this information just by looking at the diffraction pattern. You can potentially see that you can say that, okay, this is a FCC, this is a BCC. That much information you can generally get, but that is very difficult in the case of carbon, especially non-graphitizing carbons. It is very, very difficult to get this kind of information, and diffraction patterns can often be misleading. So if you even try to analyze this particular index, this particular diffraction pattern, it's very difficult to get any information, because also your even when you have pure graphite, you do not have hexagonal closed packing. You have a hexagonal structure but not closed packing, and all of these rules which allow you to find the potential crystal structure, they are for well-defined crystal structures. They are for HCP in this particular case, and our material is not HCP. So don't get too confused by the diffraction patterns. That is what I'm trying to say. Okay. Intensity profiles, they look like this. This is some green blue color. It can be any other color, because as I said that this is something that you externally plot. But what you're doing here is you take any local any region. If you see, say, stacks of a few sheets, so if you're looking at the cross section, here we have these two sheets. So in that case, you can, if you draw a line here and you plot the intensity profiles, okay, so in that case you will know where do you have your darker regions and where do you have your lighter regions or brighter regions, and then you can basically just find out using post-processing what is the separation. So here in this case you see 0.34 nanometer. Does that remind you of something? What is the layer separation? What is the d-spacing for graphite? Perfect graphite 3.3354 angstrom. Okay, so this is for perfect graphite. But if you have something between 0.335 in terms of nanometer, 0.335 and 0.344, in that case we call it turbostratic. More than 0.344 also can be considered some curved carbon structure, or you can also, if the spacing is too much, like 0.4 or something like that, in that case you will say that this is either this is not a any type of, so it should be considered a completely disordered carbon. So that is number one. Or in the case of TEM images, if you are finding the separation that is very much larger than 0.34, you might actually be looking at actually the top view and not the stacks. So you might actually be looking at something like this. So this white line and then this brown one. Rather than this, then you're probably looking at this. So this is what we'll come to. Okay, so these are the type of images that you, or type of, there are many more, by the way. Transmission electron microscopy can be used for various purposes, various types of imaging. But these are the types of images or patterns that we use generally for understanding carbon materials. Okay, so yeah, TEM patterns of non-graphitizing carbons. What I have written here is graphitic but not graphite. These kind of carbons. These are the carbons that are most extensively studied, because these are important to us for a lot of manufacturing purposes, for a lot of device application purposes. So other than carbon nanomaterials, what you study the most is these non-graphitizing carbons. Graphite, we can already get, if your graphite crystal is perfect, then you already know it from XRD. And then you probably don't need to go into very sophisticated techniques that you can learn it for understanding of the technique itself, but as such for characterization, XRD is good enough for perfect graphite, and also Raman spectroscopy will give you one good sharp peak. So what is difficult to understand, what we are trying to learn is these graphitic but not yet graphite type carbons. They can also be graphitizing carbons, by the way. Not necessarily non-graphitizing, they can also be graphitizing, but the point is that they are not yet graphite. Okay, so these kind of patterns can look very complex because well, we have multiple planes in these materials, and these planes are not necessarily, unless some of them are often turbostratic. So in that case, very difficult to find the alignment of these planes, and you're never going to get these perfect crystal planes in 3D. So these are the interesting materials, and here I have shown you one image. This is an image of a glass-like carbon, or at least the material that was prepared using a phenol formaldehyde resin at around 1200 degrees centigrade. But the film itself was extremely thin. So in this particular, when we were taking this particular image, what we did was something known as in situ TEM. In situ TEM means you heat your structure inside the TEM itself using a joule-heated substrate. Okay. You can purchase different types of, by the way, substrates for transmission electron microscopy. Typically, what you use is known as a TEM grid. So grid, as the name suggests, it has like these kind of structures. It looks like a net pretty much, but it is made of metal, and it is circular, and it's about a couple of millimeters in terms of size. Okay, so you will use this kind of grid, and then you will place your sample on top of it, and then perform the imaging. When you are performing the slicing using FIB, then you may already place your sample on top of the grid inside the FIB system. So this is how you analyze them. But you can also perform the heat treatment inside the transmission electron microscope, given your sample is extremely thin. So when I said, so you should in principle go below 20 nanometer. Why? Because well, you are going to, if let's say you're going to carbonize a polymer, then you will also have some byproducts. You know that there are some tarry materials and so on, so you don't want to contaminate this very expensive machine. So you want to make sure that the quantity of the material is as small as possible. Anyway, so this is the image of some non-graphitizing carbon. Okay, what do you see here? This is a crazy image, right? What can you say? Maybe all you can tell from this image is that this material is definitely not graphite, and definitely there is no 3D organization in the material. That's all you can say, right? You can really not tell anything about the material itself, or about how the crystallites are organized and so on. We can say that crystallites are randomly organized. Okay, that much also you can say, but how exactly are they organized? What is the crystallite size? What is the type of organization? Whether you have all the planes, these 2D planes, if we talk about whether all of them are of the same size or different sizes, whether they have the curvature or whether they are flat, none of that information is available from this kind of TEM micrograph, right? Okay, so this is for the bulk carbon materials. Okay, what about carbon fibers, tubes? There you can get relatively more realistic information, if in the case given that your fibers or tubes are thinner than 20 nanometer. Okay, if they are larger than 20 nanometer, as I said, that you can measure the diameter, but there is little information you can get about them. Okay, but samples that are thinner than 20 nanometer, there, okay, and they are individual tubes or let's say fullerenes and so on, then you can get a more realistic information, so to say. Okay. Similarly, what about graphene, single, bi, and multi-layer graphene? You can see them, yes, but it's hard to say that this is a single layer graphene. This is you should not do that unless you're really 100% sure, and you also have some other data to show that this is. So for example, Raman spectroscopy can be used for differentiating between single and bi-layers. So if you have any minor disorder or the turbostratic arrangement within your sheets, you should definitely make sure that you have some other characterization technique that tells you, gives you more information about this kind of arrangement, because in the TEM images it is hard. You're looking at this, it's hard to say what it is. It may, if you have completely these two are your graphene sheets, let's say you have bi-layer graphene but it looks like this, then you might end up thinking that this is single layer, but it is not the case. So okay, this is something you also need to understand. Grain boundaries. So TEM is also because we are trying to understand mechanical properties of the materials, right, of the crystals. So grain boundary analysis is also one of the important applications which is often used for various metals. So TEM is a very good tool for understanding grain boundaries, but again, in the case of carbon, try not to do it because you don't know where are your planes. One is here and the other one is here. So if you see some boundary, you don't really know if this is really a grain boundary or these are just two different planes at two different locations. So again, this is where you need to be careful. Okay, so here I have shown this example of a grain boundary. If you see with this this box I have shown, do you see in the back there is some sort of this broken line, and this broken line can be thought of as a grain boundary because these kind of grain boundaries have been observed in graphene-like structures. But at the same time, we can never be sure if this broken line exists in just one plane or there is some other graphene plane which is far from your original plane. But at the same time, it has some defective edges. So this is also common in the case of graphene and graphene-like structures to find edges which have a lot of these unpaired bonds, okay, which basically means that you may have the defective edge which is maybe far from your plane. So just be careful with these things. Okay, so now coming to some more structures that you can potentially see in your carbon materials, but not necessarily. So I'm just taking some very simple geometries to explain whatever I have just said. Okay. You see some circular, this ring-like structure in your carbon. So you can go back and again see that the image, the TEM image that I have shown. Okay, you see a circle, or not a complete circle, this ring-like structure, whatever it is. What are the possible 3D arrangements that you can think of? What is the 3D shape that you can think of? If I see a ring, I can think of it as a particle. I can also think that, oh, maybe it is a tube. Because I'm seeing only the 2D structure, right? So maybe it is a tube. Okay, that is possible, but it could also potentially just be a, some randomly organized sheet. So it can be something like this, which is not completely closed, right? And this is not really a tube. This is just some graphene flake which is just folded, which has a curl. Okay, so let's see. We can have various possible 3D shapes that will give you this 2D projection. Okay, so the first one is more like a tube. Again, because this I did not draw a complete tube, but it is this basically. Okay, so this is your first one. The second one, as I said, that could be some randomly rotated, randomly folded graphene sheet. Okay, and the third one can also be what looks like more like this annular structure, more like a very thin ribbon-like structure. Why I'm describing this ribbon-like structure? Because this has caused confusion. If you remember some of the microstructural models of non-graphitizing carbons, especially glassy carbon, they suggest that you have ring-like geometry, ribbon-like geometries, long ribbon-like geometries. But in principle, if you think about the fabrication techniques, if you think about the energy of the overall structure, these will be very high energy structures. These are actually there is a very low probability of finding these ribbons in any carbon materials. But the point is that you could potentially misinterpret the TEM image to think that this is a ribbon-like structure. There may also be some ribbon-like structures. Okay, so the point is that from TEM image, don't just use this technique for developing a microstructural model. Okay, but of course, if you know other things about the material, like you already know how the X-ray diffraction pattern, already know other information, chemical information, also surface property information, porosity information, whatever, you have some other information about the material density. Density becomes important in the case of non-graphitizing carbons because you may have closed porosity, right? So all of these, this information, you can integrate it then with your TEM info. Okay. What else? So some couple of these little structures I have made just for your understanding. What do you think of this? These can be 3 concentric tubes. Why not? Multi-layered, what is it called? Multi-walled carbon nanotubes. Again, I'm seeing just this view. Very well, it can be multi-walled carbon nanotubes, but it can also have some very funny structures. So I have shown this. Let's say you have these ring-like structures. then there is one over here, and the other one is over here, and then there is a smaller one somewhere here. And then there is also a complete plane in between, which you don't see because you're only seeing the top view, and the edges are not present within this structure. All of this, if you look from the top, will also give you these kind of three rings. This will be the projection. Or similarly, if you have three concentric particles, so you have these carbon particles or carbon onion-like structures, even they will look like this. So there is basically, these are one sphere, and then on top of them another on top. So these are basically particles, multi-layer. All of these are possible. So the point is that the projection itself may not be sufficient. You should know other things about the material. Some more examples. So here is another example. This is of this kind of structure you will often see. So if I have these two things here, these two planes, so let's say this is how they are stacked, and what you see is just these lines. And you often will have these lines and then also some intersecting planes and so on. This kind of structure you will often find. If I just remove the solid part from this image and I just take this, wireframe image of the exact same structure, then this is how it looks like. Now already you see it looks already so complicated. You will often see these kind of things. So these are actually real images, again from glass-like carbon. You often feel that your two planes are intersecting, while they are not. They are actually far from each other. They can be far from each other, but they look like they're intersecting. So these intersecting planes, this is also something you need to be very careful, because here the other thing is important, that what you see is just the edges and not the entire solid material. Okay. What else? This image again, I show this. I really like this image. I show this multiple times. Okay. What I thought when I first saw this image, it looks like this. It could potentially look like this. You have so these kind of these triangular junctions definitely are present in sp2 type carbons, right? They have 120 angles, and they are present. And if you read some of the very old books and so on, there has been a reasonable description of these kind of structures as well. So these kind of structures, when I saw it, I was like really happy that I saw this triangular junction. But while it can be this kind of junction, it can also be just the top view of these various hexagons. So if I'm looking at this, if you see the circle, if I'm looking at that region, what I also see, these can just be hexagons which are on top of each other, so basically there will be some triangular junction they will make. Okay, so the point is that these triangular junctions may have a depth or not, because we cannot say that from the projection. So what do you do in that case? Okay, so what we can do, so on this slide now I'm telling you how to get reliable data from TEM, or how to do a reliable interpretation. Basically you have it's not like this is a very expensive equipment, and it's not like you can never get any reliable data from this terribly expensive equipment. Okay, but there are just a few tricks that you need to make sure that you're factoring in a few things. Okay, so first of all, this is again important that you need to have other information about the material. So for example, if you want to see some closed structures, you see these particle-like structures that I showed you before in non-graphitizing carbons. Now we know that these completely spherical fullerene-like structures exist. We also know that if not completely spherical, highly curved structures definitely exist. So this we already know. So this gives you now an information about also this information can be deduced also from the density of the material, because for example these non-graphitizing carbons, they are very low density materials. So in principle they should have some porosity, but at the same time they are impermeable to gases and liquids, which means you have pores but closed pores. And closed pores also that are not really visible. So closed pores you can also not evaluate in using other porosity measurement techniques. So it's really difficult to understand them. TEM in that case has been really helpful. That is how we understood that, okay, this was a question for a very long time, that we have some sort of pores, but they are closed because the material is impermeable. So all of this information is definitely we can get from TEM imaging, but it needs to be correlated with the other information that we have about the material. Okay, so what you can do, so here I'm telling you the techniques, right? What will help you get reliable data. Sample preparation, make sure that you minimize the stresses when you're preparing the sample. Sometimes it may be inevitable, but sometimes you can avoid it. Okay. One thing that here and there I have used this trick, that there's some TEM grids that are made of, for example, molybdenum, so they can withstand relatively high temperatures. So what you can do is you can pattern, if especially when you're working with, for example, carbon nanofibers, so electrospun carbon nanofibers, or even when you're using any other polymer, you can make thin film. So this is something, what I'm saying here is valid for polymer carbonization, not for hydrocarbon pyrolysis or other methods. But for the pyrolysis process, if you could already prepare your sample on top of the TEM grid and then heat treat the entire grid. That of course will depend on the heat treatment temperature that you want to use, but if it is let's say 900, 1000, the molybdenum grid will work. So you can place the sample already on your grid and then carbonize the whole sample, which will then minimize these stresses in sample preparation. Okay, so this is something you can do. Okay. Also, by the way, if you have hydrocarbon pyrolysis, so if you are making graphene, if it is possible, if you have, so graphene is often made on copper grids, right? On copper catalysts. So if you can take a copper grid and try to already grow your graphene sheets on top of it, then you can analyze it, and that also helps you in sample preparation. These kind of things, I don't know, maybe you can use some of these things. Okay. Yeah, the stack of layers is the most important and interesting thing, because you know that all the planes, they look crazy, they just look like intersections of several planes, but this is something that you need to be careful. Now what happens if you see two planes? So I don't know if the cross section is visible, but these are two sheets, right? So if you see something like this and you are confused if it is, what you see is the side view of this stack or the top view. So of this, so let me make it white. Yeah. So if you are confused, what you're seeing is this view from here or this view. In that case, one potential thing that you can do, and this is often done, is this intensity profile. So basically, as I said, you draw a line here and you take this kind of some intensity profile that you get. Okay. Now you measure the separation. If the separation is between 0.335 and 0.36, I have written 0.36 and not 0.344, because up to 0.3, this I have written specifically because there is definitely manual error when you are measuring the separation. Because this, as I said, that this is done by software, image processing software, and also often you will manually place your cursor when you want to measure the separation between the two intensity peaks. Okay, so in that case there is some manual error, but this gives you some information up to 0.36. You know, this might be the side view. Okay, however, if this is much larger, let's say you have something like 0.5 nanometer between the two layers, then in that case there's a higher probability that you are looking at this, because you see this separation then can be this is not necessarily 0.335 or anywhere close to it. Okay, so this is one thing that you can do if you're a little bit confused. What you can also often see in these kind of stacks is that, are you getting repeatable sort of information? So if the intensity profile between two layers is 0.34, but the second one is 0.5, the third one is 0.4 again, something like that. If you have a lot of variations between the neighboring intensities, in that case, again, this may not be a stack. So if you, however, are getting 0.33, 0.34, 0.35, 0.33 again, this kind of the profile, in that case, most likely you're looking at the side view. Okay. What else? Yeah. In the TEM machine itself, if possible, use other techniques. For example, there is something known as electron energy loss spectroscopy, EELS, and as the name suggests, it is a spectroscopy. What is spectroscopy? When we somehow measure the energy, then or the loss of energy, gain of energy in the spectroscopic techniques, that's what you're doing. So energy loss, as the name indicates, that electron goes through the material, loses some energy, and that is what we measure. So the loss of energy. This also you can do. This will give you some more information. So basically within there's also something known as STEM, STEM, which gives you some idea about the depth of the structures. So if your equipment is equipped with these kind of things, then you can definitely do that. Not necessarily all the TEM setups are equipped with these additional things, because already this machine is very expensive, and also the maintenance of the machine is also very expensive. It also requires continuous high vacuum. So this is it's not something that you should definitely respect it. It's a very expensive machine, but at the same time you will not always expect that you will have all the features in your TEM. And also these features are changing every day. So if somebody purchased a TEM microscope 5 years ago, that might already not have some of the recent features and so on. So this is also a very constantly changing field, and you cannot just buy a TEM every other year, for sure. Okay, the third one that I have written is this in situ carbonization. So I will also provide citation of my own paper where I have performed this, so maybe you will get some idea from there. From that paper, I have also taken some of these images, so this is also for the citation, the acknowledgement. Okay, so this is if possible. Then make sure that you don't contaminate the TEM setup, but you can perform in situ experiments as well. Okay, further reading some NPTEL lectures by Anand Subramaniam and Professor Sankaran from IIT Madras. So there are definitely other lectures as well on microscopy. So from there you can learn the fundamentals of TEM, which I have not described here. This book, however, by Williamson Carter is one of the standard books for understanding TEM. And so the second one is my paper, from where I have taken some publication for some of the pictures. Third one is it's a review article written by PJF Harris, which gives you a very interesting history of how TEM and carbon materials have been connected, and what were some of the first carbon materials that were analyzed using transmission electron microscopy.